There is a huge spread between the lowest and highest percent risk across both breast and ovarian cancer in BRCA1 and BRCA2 gene carriers. Some of this variation can be explained by the 25 SNPs, or single nucleotide polymorphisms, linked with the risk of developing breast ovarian cancer for BRCA1 or BRCA2 carriers, or as recent evidence shows, other genes involved in the repair of double-strand breaks such as PALBS associated with the homologous recombination and inter-strand cross-link repair pathways. An excellent article appeared in this August 7th issue of the New England Journal of Medicine describing the impact of PALBS mutations on familial breast cancer. One or more SNPs in either of these DNA repair pathways, interstrand cross-link repair, single-strand DNA repair, or homologous recombination, can lead to increased risk of what we typically call BRCA1 and BRCA2 breast cancer. As this graphic clearly illustrates, BRCA1 and BRCA2 breast cancer is much more complex and complicated. Maybe in the near future we will reclassify BRCA1 and BRCA2 breast cancer based on gene mutations in one or more of these DNA repair pathways. As we will see in the next couple of slides, how the BRCA1 and BRCA2 genes trigger either breast or ovarian cancer is dependent on estrogen or its metabolites. In normal functioning cells, BRCA1 and BRCA2 are tumor suppressor genes, whose function is to maintain genomic integrity by repairing double-strand breaks or preserving interstrand cross-links between DNA base pairs in concert with a master signaling regulator called NRF2. NRF2 protects DNA from oxidative stress by enlisting the help of antioxidant genes. You may be familiar with some of these already with your understanding of the antioxidant cascade, genes like glutathione synthetase and reductase. If BRCA1 gene is mutated, it no longer partners with NRF2, leading to massive oxidative stress and cell apoptosis. However, in tissues like the breast and ovary, where estradiol levels or its metabolites are high, estradiol or its metabolites can partially turn on NRF2, keeping these cells alive, accumulating more and more mutations, and ultimately leading to breast or ovarian cancer. This slide shows the interconnection between NRF2 and the 6 different antioxidant pathways, namely production of NADPH, iron sequestration, glutathione utilization, quinone detoxification, glutathione production and regeneration, and TXN production, where TXN stands for thioredoxin, and as the name implies, is involved in cell redox homeostasis. A SNP on one or more of the genes I just mentioned, affiliated with neutralizing free radicals by different antioxidant pathways, can alter the cell's ability to survive, depending on whether the SNP either upregulates or downregulates gene expression, resulting in a greater risk of cancer in estrogen-rich tissues. Those genes I have circled in red are already being evaluated in a gene panel developed by Total Quality Medicine here in Mexico. Having this genetic information may lead to better nutrigenomic strategies to mitigate some of the effects of these gene mutations. More research will be needed, of course, but it shows the potential for individualizing and personalizing treatment strategies unique to a person's DNA map. What is clear is that genetic mutations, not only in the DNA repair pathways, but also in the antioxidant genes linked to NRF2, could contribute to a greater risk of breast or ovarian cancer in women with BRCA1 or BRCA2 mutations. This leads me to my next question. Who needs BRCA1 counseling and genetic testing? If you are a patient, then the criteria is pretty specific depending on the type of cancer, gender, and age as to when it was diagnosed. Breast cancer at age 50 or younger. ovarian cancer at any age, male breast cancer at any age, Ashkenazi Jewish descent, and has a personal history or family history of ovarian or pancreatic cancer. This would qualify for BRCA1 counseling and genetic testing. If the patient's family is concerned about the penetrance of BRCA1 and BRCA2 in the family gene, then the following criteria can be used. Remember that consulting a medical professional who may or may not refer you to a genetic counselor is also advisable. 2 breast cancers in the same person or in the same side of the family. Family member diagnosed with triple-negative breast cancer at any age. Pancreatic cancer and in breast or ovarian cancer in the same person or on the same side of the family. 3 or more family members with breast cancer on the same side of the family. Or if a family member had been previously identified with BRCA1 or BRCA2 gene mutations. This would also qualify for BRCA1 and BRCA2 gene testing and counseling.